In this video, we will discuss about altered N-Glycosylation pathways in the T cells during chronic inflammation. In our previous videos, we have discussed about this altered N-Glycosylation pathways in the innate immune cells, that is NK cells, neutrophils, and these dendritic cells. However, in this video, we will discuss about altered N-Glycosylation pathways in the T cells during chronic inflammations. And T cells plays a role either or both in the anti-inflammatory response as also in the pro-inflammatory response. And these naive T cells can be differentiated, or the lymphocytes can be differentiated into these uh, mature T cells, or these anti-inflammatory cells or pro-inflammatory cells based on its interactions with the cytokines and the antigen presenting cells. And these are the brief overviews. Has been shown that when the naive T cells interact with this interleukin 4, it will leads to the Th2, and similarly when it's interact with this TGF beta, it leads to Treg, and when interleukin 12 leads to this Th1, interleukin 6 and TGF beta, both of them when come plays a role, interacts with naive T cells, it leads to this Th17, and then in the presence of only interleukin 6, it will lead to this TFH cells. And in the case of this anti-inflammatory TS cells, these are the pool of cells that plays a role towards anti-inflammations, such as these T regulatory cells, Th2 cells. Yeah, these are the two group of cells. So in the case of these T regulatory cells, what are the changes that has occurred is that the exosomin pathway is continues to occur, that will leads to the post-translation modification or the glycosylation within the Golgi. However, this MGET pathways has been MGET5 gene has been dysregulated, and because of this, the GLICTAC additions to form this kind of structures or this kind of structures has been inhibited in the case of this T regulatory T cells. And what happens because of this? The interleukin 2 receptors will get decorated with the glycan structures that can be seen here. And that prevents the interleukin 2 to come and interact with this interleukin 2 cytokines to come and interact with this interleukin 2 receptors. So this glycan acts as a physical barrier for the interactions between interleukin 2 and interleukin 2 receptors. And because of that, this T regulatory cells plays a role towards anti-inflammations, and this interleukin 2 will not change this anti-inflammation cells into the pro-inflammation cells. And now when it comes to these pro-inflammatory cells, there are different kind of N-glycosylation alteration occurs compared to the anti-inflammatory TS cells. So in the case of pro-inflammatory TS cells, the example is shown here about this Th17 cells. So in this case, what happens first? This exosomin pathways has been dysregulated in the case of Th17 because of the interaction with this Th17 cytokines, and this within this exosomin pathways, this glucose 6-phosphate to glucosamine 6-phosphate will be dysregulated because of the dysregulations of this GFT1 gene, and what it will leads to and it will ultimately leads to this dysregulation of this UDP GLICNAC pathways, and because of that and this UDP GLICNAC is essential for the N-glycosylation or O-glycosylation pathways in the Golgi and also in the endoplasmic reticulum. So because of the dysregulation of this UDP GLICNAC, further post maturation of the glycosylation process or the post translation modifications get dysregulated within the Golgi, and along with that, the other glycotransferase enzymes also get dysregulated, such as MGET5 here is also get reduced or down regulated, whereas ST6-GAL1 and FAT8 get upregulated, and because of that it will leads to the T cell receptors to be decorated with the glycan structures in a way that it will make the T cell receptors hyperactive, and this is why we call these cells as activated T cells. This is one of the reasons. At the same time, for this CD43 and CD45 will get decorated with the glycans that will prevent them that will prevent this interaction with this galectin 1 and galectin 3 that acts as a inhibitor for the active that acts as an inhibitor for the T cells. And also this SIGLEC receptor SIGLEC22 for examples also act as an inhibitor for the T cells. So when the cells are decorated, the T cells are decorated with the glycan structures, this it will also prevents the T cells interactions with these SIGLEC receptors. For example, these non-sialic acids glycan structures will not be able to interact with these SIGLEC receptors. Thereby, the possibility of these activated T cells to get down-regulated will get reduced to a greater extent. One of the reasons is because of the change in the glycosylation process that occurs in this pro-inflammatory TS cells. So I hope you got a basic idea. So this is just overview, and that will, and one more thing is this about this hepatic ST6 galvan. This hepatic ST6 galvans will get reduced in the cellular matrix because it interacted with this, both the interleukin 2 receptors and also the CD45 receptors. That will lead to the down regulation of this ST6 galvan. So I hope you got a basic idea or preliminary ideas about this altered N-Glycosylation pathways in the T cells during chronic inflammations, both in the anti-inflammatory TS cells and the pro-inflammatory TS cells. And this is panel B is just the basic informations how the naive T cells get differentiated into the different populations based on the interactions with these cytokines. If you need to understand more about these altered glycosylation pathways in T cells during chronic inflammations in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.